Hi, it's me, Teresa, and this lecture is going to be about gustation, your sense of taste, olfaction, your sense of smell, and then at the end I'm going to talk a little bit about somatosenses. Alright, so the first of these three senses that I'm going to talk about is gustation. Gustation and olfaction together are called your chemical senses. And the reason that they're called your chemical senses is because there are certain chemicals in the world that interact with your receptor systems in order for you to experience these senses. So when we were talking about vision, we were talking about light information. And your photoreceptors in your retina transduce light information. When we were talking about hearing and audition, we were talking about sound information. And it's your auditory hair cells that transduce sound waves. And so you experience sound. When we're talking about gustation and olfaction, taste and smell, there are actual chemicals, molecules, in the world that go into our mouth and into our nose, and these chemicals interact with receptors there. And so that's why these senses are called your chemical senses. So first I'm going to talk about gustation, and I'm going to go through the basic tastes, which are salty, sour, bitter, sweet, and umami. Umami, you can see spelled there at the top of the board. Umami means savory. It was Japanese researchers who discovered the umami receptor, and so they named it the umami receptor, which loosely translated means savory. So I'm going to talk about all of those, and then I'm going to talk about the names for the cells, which are TRCs, taste receptor cells, and the chemicals that are received by taste receptor cells are called tastants. So any chemical that is in your food or any other substance that you can taste is called a tastant. And then I'm going to talk about how a taste bud is structured and how the little bumps on your tongue called papillae are structured. And then I'm going to talk about the transduction process itself very briefly, and then the pathways in the brain. So there are 5 basic tastes that we can experience with our sensory equipment. And these 5 basic tastes correspond to certain chemicals that are in our food or any other substance that we're tasting. And those taste receptor cells, TRCs, are salty, sweet, sour, bitter, and umami. And there are all the different kinds of taste receptor cells, all 5 of the basic tastes, located all over your tongue, though there are higher concentrations in certain areas. So you may have heard people say things like, you taste sweet foods with the tip of your tongue, or you taste bitter foods with the back of your tongue. And the reason that people say that is because there are higher concentration of bitter TRCs at the back of your tongue, and there are a higher concentration of sweet receptors at the tip of your tongue, but it's just a higher concentration. There are all the different kinds all over your tongue. And the bumps that you see on your tongue when you look at your tongue, you may have called them taste buds, but those actually aren't taste buds. The bumps on your tongue, okay, so here's the tongue, and then we blew up one of the bumps so that you can see its structure. That bump is called a papilla. Okay, so you see papilla written at the top of the board, or multiple is papillae, so with an E at the end. So the bumps on your tongue are called papillae. And those are the things that you can see when you look at your tongue. Here's an electron microscope picture of one, and there's a drawing of one. And the taste buds are actually down inside the papillae. So inside the folds of the papillae, which you see up there, the taste buds are actually inside the crease of the papilla. Okay, and so you see the papilla there in pink, and then the taste buds are the little blue spots. And so a taste bud is blown up right here. And a taste bud, like I say, is kind of embedded inside the papilla. And a taste bud contains 50 to 150 taste receptor cells, TRCs. So each of these cells that you see here is a taste receptor cell. And each one responds to a different tastant. And so when a chemical comes into your mouth, let's say it's a lemon, and so it's sour, okay? You taste the lemon and it's sour. If the taste receptor cell is one for sour, the tastant, the sour chemical, the chemical that creates sour, will bind up here at the top of the cells, the gustatory receptor cells, or the taste receptor cells, okay? And so the taste bud is kind of like a bunch of bananas, and each of the bananas is a taste receptor cell, okay? So the taste bud is what houses the taste receptor cells in the same way that the retina houses the photoreceptors or the cochlea houses the auditory hair cells. The taste bud holds the taste receptor cells, the TRCs. And they are embedded inside the papilla, like you can see there. You may hear the word flavor applied to food. And flavor is a little bit different than taste. Taste is confined to those 5 different basic tastes. So tasting is actually done with your mouth, okay? And you have taste receptor cells, as you know, all over your tongue, but also even in your cheeks and in your palate at the top of your mouth. There are taste receptor cells all over your mouth. And those 5 basic flavors can be transduced by your taste receptor cells. But you may be thinking, well, I can taste a lot more things other than just sweet, salty, sour, bitter, and savory. I can taste a lot of other flavors, really complicated flavors. And that's because flavor is taste plus smell plus touch. So flavor consists of not just the taste of a food, but also the smell of the food and the way it feels inside of your mouth. So all of those things go into flavor. All right. So at the very tips of your taste receptor cells are little microvilli. Okay, little cilia, little hair-like extensions, okay? So they call them a lot of different things. They'll call them microvilli, that just means little tiny finger-like projections. Or cilia, you know, again, just little projections. So gustatory hair, however you want to call it. It's very similar to the stereocilia on auditory hair cells, okay? So kind of similar in structure. And so all of those little microvilli form what's called a taste pore, okay? It's a taste pore. It's a little spot where the tastant can sit so that it can be tasted, so that the cells can respond to this chemical, okay? So the chemical goes into the taste pore, and then the taste receptor cell is going to respond to that chemical, to that specific chemical. And the processes are slightly different for the different kinds of taste receptor cells. So for salt, when you're tasting salt, and you put salt onto your tongue, it's going to be the same beginning for all of these things. You're going to have whatever the tastant is. It goes inside your papilla into the taste bud, and it binds at the taste pore, at those little microvilli on the tops of the gustatory receptor cells. And then it's going to change the activity of those cells. Okay, the transduction is done by the taste receptor cells. And you can see here they have dyed the tongue, and so you can see the papillae on an actual tongue. Okay, when salt comes into the taste pore, when you taste something salty and it comes into one of your taste pores, and it's a taste receptor cell for salt, the salt actually contains sodium, okay? And so the sodium itself can go right into the ion channels. So salt is a chemical that contains ions that your body can process. And so when salt is on your tongue, it's as simple as the sodium coming into the ion channel and depolarizing the cell. And that's as simple as transduction in a salt receptor. Okay? For bitter, it's a little bit more complicated. It's a metabotropic receptor. So the salt receptors are ionotropic, and there really isn't even a neurotransmitter involved at that point. The ion itself is directly going into the channel. And then it is going to be that cell that then has a cascade of responses that changes the amount of neurotransmitter it's releasing. For bitter, it's a metabotropic process. And so when you take bitter, flavors are like, because sometimes people get bitter and sour mixed up. Sour is like a lemon, or a lemon patch kid, or a sour patch kid, or sour candy. Whereas bitter is like, you know how raw broccoli tastes, or Brussels sprouts? Okay, that's a bitter taste. So they are a little bit different. But I'm not going to have you memorize the metabotropic process, just that, salt is an ionotropic thing where the sodium comes right into the ion channel. Bitter is a metabotropic receptor with a G protein and an alpha subunit. And then the other three, salty and umami, and, hold on a second, well, sour is also kind of simple, okay? So I'll talk about sour just a little bit. In sour foods, so like lemons and Sour Patch Kids, they have hydrogen ions in them, okay? And hydrogen ions are released by these foods, and it blocks a potassium channel. And when the hydrogen ions block the potassium channel, the positive charge builds up inside the cell, okay? So you've got your salty receptors where the ion comes directly in. You've got your sour receptors where there's hydrogen ions in the sour food that block a potassium channel and let potassium stay in the cell so it gets pushed up towards action potential, or it gets excited. And then there's bitter, which, like I say, is a metabotropic process. And umami and sweet are also metabotropic receptors, and they involve a second messenger system, and they're more complicated, so I'm not going to have you memorize those steps or anything, okay? You just want to know that regardless of what the tastant is, okay, whether you're talking about any of the 5 basic tastes, they go into the sides of the papillae where the taste buds are, and the taste buds are clumps of taste receptor cells, and depending on what kind of tastant it is, it's either an ionotropic process or a metabotropic process. Different different animals have different taste systems. And part of it has to do with the receptors themselves. So for example, a cat does not have any sweet receptors. They don't code for sweet receptors. Cats didn't evolve to eat anything sweet. They didn't evolve to eat fruit or anything like that. So there was no reason to have a receptor for sweet foods. Humans, we evolved to eat fruit and things like that, so we have receptors for sweet foods. Now you might occasionally find a cat that likes different kinds of fruit. Like I had a cat that loved watermelon, but they can't taste that it's sweet. They just like the texture or something else about it. It's not that they like the sweet flavor because they can't taste the sweet flavor. So, color vision, right, depends on the kinds of photoreceptors you have, the kinds of cones you have, and your flavor, your taste perception depends on the kinds of taste receptor cells you have. But there are other things that happen that also influence how different organisms experience taste. So for example, human beings, if you smell or taste poop, feces, it's terrible. We hate it. It's processed as very unpleasant. And that's because it's toxic to us. If you eat poop, you can get really, really sick, okay? So we evolved to have a dislike for that. But a fly eats poop, right? Flies eat poop. So flies aren't grossed out. They don't have a negative experience of poop. They're attracted to it, right? And that's about other things going on in the nervous system. The reason that humans are grossed out by poop and like, sweet foods like candy is because our brains process one as gross. It activates parts in our brain that say, oh, that's gross. And the other one activates parts in our brains that say, that's delicious. So now I want to talk about what those pathways are that go back to the brain. Okay, so these pathways are going directly from your mouth into your brain. They don't go through the spinal cord. They go directly from the head into the brain. And so it involves cranial nerves. And I listed the cranial nerves right here that are involved in taste perception. So your facial nerve receives information from the front 2/3 of your tongue, okay? So the front 2/3 of your tongue, they send their taste receptor cells, synapse onto gustatory nerve, so that you can taste. And for the front 2/3 of your tongue, that's the facial nerve. From the back of your tongue, the hypoglossal nerve receives information and sends it on to the brain. From your back of your throat, towards your throat, and from your soft palate, okay, so from the top and back of your mouth, that's where the glossopharyngeal nerve communicates information directly into your brain about the taste of your food. But the trigeminal nerve, that doesn't send taste information. That sends temperature information and information about like the texture of the food, okay? So not about the taste, but about how hot it is or cold it is, how chewy or crunchy it is, and that kind of information. Not specifically taste information, but the other kinds of somatosensory information you can get from your mouth. Because your mouth it doesn't just taste things, it also feels things, right? Okay, so that's your trigeminal nerve. Regardless of what nerve you're talking about, the first place in the brain that information is going to go is your medulla. And the medulla is part of the brain stem. It's part of your hindbrain, part of the lowest part of your brain, okay? Your medulla. And so it processes really basic information And our chemical senses actually are a lot of lower order animals, a lot of animals that don't even have a cortex, respond to taste information. So for example, let's say it's let's say it's an alligator, okay? An alligator bites into a fish and it tastes good to the alligator. And so, you know, it eats the fish. Alligators have a medulla, okay? They have a brainstem, they have a hindbrain. And so that information can be processed there for even lower order animals to just know, oh, is this, you know, a positive flavor or a negative flavor? Is this something that I should be eating or not eating, okay? But then the second stop for the information is the thalamus. And you shouldn't be surprised because the thalamus is the main routing, you know, it's that relay station in the brain. And so the next place we got to pass the information is to the thalamus, so that the thalamus can send the information to the correct part of the cortex. And the correct part of the cortex for taste information is somatosensory cortex. Okay, so that's where you're going to go next, up in the parietal lobe. So you have projections from the cranial nerves directly into the medulla, from the medulla up to the thalamus, and then from the thalamus onto somatosensory cortex. And that's where you process gustatory information. Okay, so now I'm going to talk about your sense of smell. And the technical word for your sense of smell is olfaction. And the receptors for olfaction, the cells that do the transduction, are called your olfactory receptor cells, or ORCs. Okay, so the transducing cells for taste were called TRCs, taste receptor cells. And so for olfaction, they're called ORCs, olfactory receptor cells. And the place where they are is way up in your nose, called the olfactory epithelium. Okay, so I'm going to talk a little bit about the olfactory epithelium, which is written right there. And I'm going to talk about the different kinds of receptors. And in the same way that a tastant was a chemical that binds to your taste receptors, an odorant is a chemical that binds to your olfactory receptors, okay? It makes an odorant. And then I'm going to talk about the pathways that happen after that, getting back to the brain, okay? So you have over 20 million ORCs. You have over 20 million olfactory receptor cells up inside your nose, okay? So here's your nose hole, here's your nostril, and here's the sinus behind. You know, you've heard of having a sinus infection, okay? That's where the tissues up here in your sinus get all swollen. And that makes it so that you can't smell very well, because at the top of your sinuses, at the top of your nasal cavity, is what's called your olfactory epithelium. Or some people call it your epithelial lining. Some people call it your olfactory mucosa because it's covered in mucus, okay? It's wet. It's wet up there. You make mucus in your nose, obviously. So your nasal mucosa, your olfactory mucosa, your olfactory epithelium, whatever you want to call it. But that's your olfactory epithelium, and it is what houses, what holds the olfactory receptor cells, the ORCs. So in the same way you had like the retina hold the photoreceptors, you have the olfactory receptor cells inside the olfactory epithelium. And like I say, there are over 20 million of them total, but there are at least 350 different kinds. So when we were talking about taste receptor cells, there were only 5 kinds. For smells, you have over 350 different kinds of receptors for different kinds of smells. And their little receptors, the place where the chemical is going to bind, is up here in the olfactory epithelium. Here are the ORCs, and their little dendrites are poking out into the top of your sinus cavities. And when you smell an odor, okay, so let's say like somebody's cooking bacon, little molecules of the bacon travel through the air, okay? They're called odorant molecules. Little actual molecules of the bacon float through the air and go into your nose and go up into the olfactory epithelium and bind to the receptors on the dendrites there. Just like a neurotransmitter would bind to them, okay? But again, these are your chemical senses. So these are chemicals that are binding to those receptors. They just bind right to it. And those receptors, so here's the receptor cells, the olfactory receptor cells, their axons go through, so at the top of your sinuses is bone, is your skull, okay? So this is your skull bone right here. And their axons go through the skull bone, and on the other side of the skull bone is the olfactory bulb. And that is the first place in the brain where smell information goes, where olfactory information goes. And there are, like I say, over 350 different receptor types. They're metabotropic. They're all metabotropic. Usually a second messenger system opens up a sodium channel and lets sodium in, and that pushes the cells to action potential. But there are other mechanisms too, and you don't have to learn them for the purposes of this class. But what you do have to know is that regardless of what the odorant molecule is, it is an actual molecule of that thing. So if you smell like poop, it's little poop molecules going up and into your nose, binding to the receptors, and then causing a metabotropic process to happen that excites those cells. So here are a whole bunch of different kinds of olfactory receptor cells pictured in the olfactory epithelium. Okay, this is the olfactory epithelium, the mucousy lining at the top of your sinuses. And here's the olfactory receptor cells. Here's their dendrites. Here's an odorant molecule. It can bind to the appropriate dendrite. And then the axons, like I say, go up into the olfactory bulb. And the olfactory bulb is made of a bunch of clusters of cells, a bunch of clusters of cell bodies with their dendrites connecting to the end feet of the receptor cell. So the receptor cell sends its axon up into the olfactory bulb, and it sends its projection to one of these little nodes that are, each one's called a glomerulus, okay, written right there, glomerulus, or plural is glomeruli, okay? And what happens is you have all kinds of different, like I say, 350 different olfactory receptors. So you might have one over here that's for bacon, and then here's another bacon one, and then here's another bacon one. But then this one's for roses and roses, and roses over here. And then another one responds to cinnamon, let's say, okay, all the different smells, okay? So the ones that are for the same smell, so like let's say it's all for bacon, okay? All the bacon ones send their axons to one glomerulus. And then the other kinds send their information to different glomeruli. So depending on which glomerulus is activated, you're detecting that certain smell, okay? So all the, let's say, I'm just making up imaginary smells like bacon or poop or roses or whatever, okay? It triggers its specific kind of olfactory receptor cell, and then it sends its information back to a glomerulus, and all of the other ones like it send their information to that glomerulus too. And then it's the pattern of activity of the glomeruli that your brain interprets as different smells, okay? So let's say you're wine tasting and you have a Merlot, And it activates certain olfactory receptor cells, certain ORCs, and they send their information to the appropriate glomeruli, and that's how you know, oh, this is a Merlot, okay? But let's say that next you want to taste a Pinot Noir, okay? It's gonna share some of the same glomeruli, but also activate other different ones, and that's how you tell the difference between two different tastes, okay? So, steak is gonna have one pattern of activity, and cheese is gonna have a different pattern of activity, and all the different smells make different patterns of activity in the glomeruli, and that's how you know what the different smells are. After the information is translated as whatever the smell is by your olfactory bulb, the olfactory bulb sends projections back directly to your limbic system. And your limbic system includes structures like your amygdala, okay? And so that's why our sense of smell can be highly tied to emotion. Because these projections, they're being first processed, what smell is it, is processed in the olfactory bulb. But then the limbic system tells you any emotional connection you might have to that smell. So that's why, like, if you smell your grandma's cooking, that's why it makes you feel good. Or if you smell the cologne of somebody you hate, you feel bad, okay? So we have these connections with these memory connections, emotional memory connections with certain smells because of the direct link from the olfactory bulb to the limbic system. And then it sends that information out to multiple other endpoints in the brain. Okay. The last thing I want to talk about is related to our sense of smell. But there's a question in people of whether it's actually another sense, okay? So when we were talking about like echolocation, okay? Bats and dolphins can do echolocation. We can't echolocate, at least not very well. We can do some moderate echolocation, but we don't have a full organ for it the same way that a dolphin or a bat does, okay? So the same kind of thing is going on when it comes to certain odorant molecules that we call pheromones. Okay, so pheromones are chemicals that are secreted by an organism, and they use these chemicals to communicate with other members of their species, all right? So, when a dog marks its territory, okay, with urine, it is putting a smell on that thing that other dogs can then smell and find out a lot of information about the other dog. Like, is that dog in heat? Is it a female? Is it a male? Is that dog old or young? Okay, there's a lot of information communicated in that smell. And so we call that communication intraspecific. That means within a species, okay? So pheromones are all about species communicating to other members of their species. And we see this very clearly in certain animals, but we're not sure about it in people. We're not sure how much pheromones influence us, how sensitive we are to pheromones. It's a current area of active research. So should we call, for humans, should we call pheromone perception its own thing, or should we just say it's part of smell? Whereas for other animals, it really definitely seems to be its own thing, okay? And let me explain to you how we know this. All right. So, first of all, when we look at the behavior of other animals, we can see that they are getting important information through their sense of smell. Like dogs are always sniffing each other to find out information about one another. Well, it turns out if you look at these animals and you were to look inside their skulls, you would see that they have an organ, a sensory organ called a vomeronasal organ, or VNO, vomeronasal organ, VNO, okay? And it is a large, noticeable structure inside certain animals' heads, okay? So if you were to look inside a rat, you would see a large, noticeable VNO. But in humans, we're not even sure if we have a VNO, okay? Animals that we know have a VNO, they have a distinct duct, okay? The same way we have nasal ducts that bring air into our nose, they have another duct that brings air into the VNO. We don't really have that. We have a little area in our nose that kind of has a pit, like a little opening, a little short hole, okay? And we might have a little tiny VNO in there, but that's up for debate. Different people think, oh yeah, it's definitely there, and other people think, I don't think it really, they call it vestigial in people, meaning it's no longer used. It's just a remnant from our evolutionary past. Okay, so there's some people who say the VNO is vestigial. It's just a remnant of our evolutionary past. Okay? So, regardless though, in animals, we can detect it clearly, and they do behaviors that indicate they're using their VNO. This behavior is called phlegmon, and you see it in lots of different mammals. And what they do is they kind of squinch their nose up, and what they're doing is they're blocking off their nasal passages and opening up the VNO duct, opening up the VNO passage. And then they kind of breathe air in to force the air up into the VNO. So it looks like, you see in the pictures, they kind of go, and they draw air in and force it up into their VNO. And this can help them, find a mate, or, know where other animals are in their territory, and this kind of thing. So we see this distinct behavior, and we know that they are, sending odorant molecules from other members of their species up into their VNO so that they can find out information, sensory information. The three different kinds of pheromones that we know of are called releaser, primer, and modulator pheromones, okay? And they do slightly different things. A releaser pheromone, like let's say a member of your species emits a releaser pheromone, okay? They emit a releaser pheromone. When another member of the species receives the releaser pheromone, it forces the animal to do a certain behavior. It triggers a certain automatic behavior. And I'll give an example. Termites use releaser pheromones, okay? When a termite is flying through the air, every once in a while it poops, okay? It's a termite, it's a living thing, it has to poop, okay? So they poop. When another termite flies over that exact same spot, pheromones from the other termite's poop hit this termite, this new termite, and trigger it to release its own poop. So as soon as that pheromone hits that termite, it releases its poop. And that's how a termite mound gets built. By pheromone communication with the termites. Ants also use these. Then we have a second kind of pheromone, and it does something a little bit different. It's called a primer pheromone. And it's kind of like a releaser pheromone, except for it triggers behaviors at a specific point during development. So, a releaser pheromone, anytime the termite smells that releaser pheromone or detects that releaser pheromone, it's going to react. With a primer pheromone, it's going to trigger certain behaviors during development. So an example is like this. Mammary glands, okay, in mammals that make milk, they secrete pheromones that attract the babies to eat. Okay, it's a smell that smells good. It's a pheromone that smells good, or is perceived as good by the babies, and so it triggers nursing behaviors. Okay, that's an example of a primer pheromone. As far as we know, humans do not use releaser pheromones, but the one that we are really interested in people are modulator pheromones. And modulator pheromones, the way that you can think of them is not that they trigger a certain behavior, okay? Because the other two trigger a certain behavior. But modulator pheromones might affect the mood of the recipient, okay? So you might have a pheromone that makes the recipient more receptive or less receptive, might change the mood of the individual. And these are the ones that we are always looking to explore with people, because we want to know, are people releasing some kind of pheromones that changes the mood of other people? And there's some evidence that human beings and other primates might use modulator pheromones, but not releaser pheromones, and primer pheromones only during development, like I say, because that's what a primer pheromone is. All right. So, the question then becomes, how much do odors matter to human beings? How much, whether you're calling it a pheromone or you're calling it an odor, obviously odorant molecules, obviously these molecules that waft through the air and go into our nose, they do affect us. You know that. You know there are smells that gross you out and smells that you love. Okay, so we know that odors have something to do with human behavior. And like I say, we know that there are certain things that we secrete ourselves, so those would be pheromones, odorant molecules that we ourselves secrete that affect the behavior of other humans. Now, are we using our VNO? That we don't know. Or is it just we're using the rest of our smell equipment, the other 350 receptors? Did they interact with pheromones? We're not really sure. But what we do know is that things like your menstrual cycle affect your sense of smell and how you process smells. So when women are ovulating and they smell male smells, they find male smells more attractive when they're ovulating than at other times during the menstrual cycle. So the way that they do these experiments is they just get they get people, men and women, to wear shirts all day long, and then they take the shirt and they put it in a Ziploc bag, and then they take all these shirts and they have people smell them and say, how enjoyable it is. Is it, on a scale of 1 to 10, how enjoyable is this? On a scale of 1 to 10, would you be, how attracted would you be to a person who smells this way? Okay, so they'll ask that. And women in general rate male shirts, the smell coming off of male shirts, as more enjoyable when they're ovulating. And that might be to encourage mating. We also know that human beings tend to, they've done the same kind of tests with people who are all different, body sizes. And it's, people are blindfolded when they're smelling it, so they can't see the shirt. All they can do is smell it. And then they rate on how well they like it. And the more obese people are, the less people tend to like the smell. We don't like the smell of our relatives. especially relatives of the opposite sex, okay? And the reason for this is because of something called, it's tied to something called our major histocompatibility complex, okay? Major histocompatibility complex. You can just call it the MHC, okay? And that is about your immune system, okay? Your major histocompatibility complex is essentially how your particular immune system is built. And people tend to like the smell of people who have a very different major histocompatibility complex than their own. Now, you're going to have a similar major histocompatibility complex to your relatives. Obviously, you guys have the same genes, and so the major histocompatibility complex, the immune system, is shared from, within a family. It's passed on by genes. And so there's something about people who have the same major histocompatibility complex as us that we don't like their smell. And those tend to be our relatives. And so we don't like their smell, so we stay away from them. Whereas people who have a very different major histocompatibility complex, they have a very different immune system, and we tend to be attracted to people who have a very different MHC than us. And they tend to be not related to us because they have a very different set of genetics than we do. And the idea is that if you bring two people who have two very different immune systems together, then their offspring can have an even better immune system because they get the good stuff from each, the good different stuff from each of the parents. And if you are not attracted to people who have the same MHC as you, their smell grosses you out, you're less likely to mate with a relative. And obviously mating with a relative is bad. It creates inbreeding, and then we have, more genetic problems with the organism. There was a researcher who believed, she was researching, I think in the 1970s is when she started her research. She believed that women emitted pheromones when they were menstruating, okay, when they're on their periods. And that these pheromones could be detected by other women, and that if you were around each other enough, detecting these pheromones in each other would make it so that your periods synced up, okay? And this is a common belief. My cousins, for example, strongly believe in what we call menstrual synchrony. And this is the idea that when women live together, they sync up their periods. Now we know that that occurs in the real world. We know that there are animals that all go into season, all become able to mate and produce offspring at a specific time of the year. But that's not true with humans, okay? Humans can mate at any time. And when she did her research, she did it on college students living in the dorms, and she found that there was menstrual synchrony, that the women in the dorms would sync up their periods. Well, since then, other researchers have looked at, number one, her own data, and then number two, have conducted lots of other studies. And at this point, there is no evidence that menstrual synchrony is a real phenomenon, okay? They went back and looked at her data, and it was just an artifact of the way she did her statistical analyses. And if you did the right statistical analyses, there was no relationship between when different women had their periods. But what does happen is something we call a confirmation bias. Meaning, if you're on your period and you notice another person on their period, you go, wow, we're synced up. Whereas if another person is having their period at a different time than you don't really notice it. So it turns out that menstrual synchrony is not supported by the science, and it's really a confirmation bias within us. Okay, that's all I have to say about gustation, olfaction, and pheromones as possibly part of olfaction or maybe its own sense in certain animals. But I do very briefly want to talk about somatosenses. Let's put that all up there. Okay, so what I want to say first of all is you really should look at your book about somatosenses. And in particular, there's parts of your book where there are like charts, like lists and charts, tables, that talk about all of the different kinds of somatosensory cells you have, because there's a lot of them. Make sure that you have good notes on that section, okay, because it is going to appear on the test. But I'm going to just very briefly talk about somatosenses right now. Your somatosenses are your body senses. So that's your touch information, hot and cold information, the stuff you're feeling with your skin and with your, insides too. Your neurons inside you that can detect this kind of bodily information. And what you have is a lot of different kinds of neurons in your skin and inside your organs too. And these different kinds of neurons interact with the world and transduce the information from the world in a bunch of different ways. There are some that have mechanical receptors where it just opens an ion channel. There are others where chemicals on your skin might interact, and then hot and cold things interact with different receptors. But it's all, you have dendrites for all of those things. You have cells for all of those things in your skin. And your touch and your pressure neurons, they show adaptation a lot like visual, like your visual neurons do. So your visual neurons get tired after a while. They will, so you know how if you stare at a light for a long time and then you look away from that light, you can still see the visible persistence of that light, like you can still see the light? That's because your neurons have become adapted to the light and they're firing in response to the light, and then when the light's no longer there, they are still, your brain is still interpreting that information. It's still sending signals for a little bit after the light is gone. And that's why you can keep seeing light even when you're not looking at it, okay? Like you look at a light and then you look away and you can still see it there, okay? That's adaptation. Your neurons adapt. And your touch and pressure ones do the same thing. So for example, when you first put on your clothes, you feel yourself put them on, but then you don't feel it anymore. And the same thing with touch and pressure. You have different concentrations of somatosensory receptors in different parts of your body, okay? So like on your fingertips, you have a lot of sensory neurons. Your mouth, your eyes, your genitals, you have a lot of sensory neurons. A high concentration. Whereas in other places, like your back, you don't. And so they call a test to see how dense your neurons are. They call it a 2-point discrimination threshold. They take 2 points and they want to see how far apart do those 2 points have to be before you can tell it's 2 different points. And if you do it on your finger, the 2 points can be really close together and you can still tell the difference. But on your back, the 2 points have to be a lot further apart before you can tell it's 2 different points. And the reason for that is because you have way fewer receptors in your back than you do in your fingertips or your lips or your eyes. You have at least 6 different kinds of receptors just for temperature. So for cold, for hot, and then there's different kinds of cold and hot. So you know the difference between spicy hot and like temperature hot, like it's been heated with, in the oven for too long, okay? Those are 2 different kinds of hot. Cold, there's icy cold, and then there's like minty cold. So you have different kinds of thermoreceptors for all those different things. Your pain receptors, you have different kinds of pain receptors, and they are actually, your experience of pain is highly influenced by your thoughts. You can use higher parts of your brain to, what they call, close the pain gate, to turn down your experience of pain. I did it when I hypnobirthed my second child and started to hypnobirth my third child. So with my second child, I went through this long process of learning how to hypnotize myself that they call hypnobirthing. And what you do is you hypnotize yourself into a real deep state of relaxation, and you hypnotize yourself to reinterpret the feelings of childbirth not as pain, but instead as something else, like stretching or unfolding, okay? So you learn to reinterpret pain. And it is extremely effective for people who can do it. So I didn't believe it was going to work. I didn't believe it at all. But I went through my entire labor with my second child, and it was over 24 hours of labor. I did not feel pain until she was crowning, until her head was getting ready to come out. And that was the first time I felt pain during the entire labor process. Most women are screaming, for several hours during the labor process. I was completely calm and experiencing no pain until she was crowning. And even then it was very mild pain that I was experiencing. And that's supposed to be like one of the most painful things a human can do. But I learned to use my cognitions to suppress those messages and interpret them in a different way. You do have internal receptors in your organs and your joints, and this allows you to, tell how heavy something is, or, so when you're picking up something and it's heavy, there's a lot more stretch of your tendons and joints. And so that's how you can tell, by that stretching, how heavy something is. And then lastly, the last thing I want to mention are your vestibular senses. So next to your cochlea, you have these three semicircular tubes that make up your vestibular system. And they're full of a really thick jelly-like substance. And when you move in space, those three semicircular canals compare information, about which receptors are being activated, and it tells you, am I right side up or upside down? Am I on my side? That kind of information your vestibular system tells you. And when you drink alcohol, the alcohol poisons the fluid inside your vestibular system. And so it can send a message to your brain that you're spinning when you're really not spinning. And so that's why you get the spins sometimes when you're drunk, because you've poisoned your semicircular canals. So drink a lot of water and try to flush your system as much as you can. Other than that, you just have to wait. But that's what's happening when you get drunk and get the spins. So you should also look at your book for the information about the vestibular system and like the components of it and the receptors in there too, because there will be a little bit of that on the test. Okay, so that's all I have to say about that, and good luck on your exam, and thank you for watching and taking good notes.